All right, this is the next lecture where we're going to talk about metabolism. If you haven't already, please watch the lecture on basic energy concepts. Before we get into the next topic, which is fermentation, I wanted to talk about types of catabolic processes. These can be divided into 3 different areas, and most of the organisms on this planet will fall into one of these 3 categories. First of all, you have photolithoautotrophs. They get their energy from light, photo, their electrons from inorganic sources, litho, and their carbon from CO2, autotrophs. Chemoheteroorganotrophs are the second subclass. They get their energy from the oxidation of organic chemicals, they get their electrons from organic chemicals, and they get their carbon from organic chemicals. So all 3 of them come from organic chemicals. Many organisms fall into this category, including you. A third class of organism is chemolithoautotrophs. They will oxidize inorganic chemicals for energy, chemo. They will obtain electrons from inorganic compounds, litho, and they will get their carbon from CO2, autotrophs. This is originally thought to be an interesting but small category of organisms. In recent decades, we've realized that chemolithoautotrophs are quite numerous. Okay, what are the systems for generating energy? Well, first of all, you can get your energy from substrate level phosphorylation. This is the synthesis of ATP from ADP, and it's coupled with the breakdown of high energy organic substrates. The energy of the chemical bonds in the organic molecule is directly transferred to ATP or ADP, forming ATP. The second type of energy generation to make chemical energy is membrane system phosphorylation. And this can be broken down in a number of different ways, and there's 3 categories: oxidative or electron transport level phosphorylation, where oxygen is the terminal electron acceptor; anaerobic respiration, that uses something other than oxygen as the terminal electron acceptor; and then photophosphorylation, that actually uses light to generate energy in the electrons, to boost the energy of the electrons, and then runs them through an electron transport chain. All right, so those are the systems. That's why you can classify organisms: photoautolithotrophs, chemoheteroorganotrophs, or chemolithoautotrophs. Those are 3 very common categories. And then ways of generating energy by substrate level phosphorylation and by using a membrane. Right. Before we get into fermentation, let's talk about glycolytic pathways, because this is a very common pathway that organisms use to break down sugars and glucose. In glycolytic pathways, the glucose is broken down to smaller molecules, and there's 2 that we're going to talk about. There's the Embden Meyerhoff Parnas pathway, and many people will refer to this as glycolysis. There are many bacteria in archaea that do this, and then there is the Anter-Deuteroff pathway. And there are other organisms that do this. Most of these are strictly aerobic bacteria that then use electron transport level phosphorylation. And one group of organisms that does this is Pseudomonas. All right, so glycolysis, the Embden Meyerhoff Parnas pathway. You've seen this before in biology because this is the process that takes place in our mitochondria. Glucose is first phosphorylated 2 times to make glucose 6-phosphate. There's an isomerization to fructose 6-phosphate, and then another phosphate is added. Now, first of all, I don't expect you to memorize these pathways. That's not what this class is about. But I want to expose you to the chemical conversions that happen. So why do you add 2 phosphates if you're trying to generate chemical energy? Well, glucose is actually a surprisingly stable molecule. And by converting it to fructose and then adding 2 phosphates, fructose 1,6-diphosphate is a more unstable molecule, and it's more able to be attacked by the next enzyme, aldolase. The next step in the EMP pathway is splitting this molecule, and aldolase splits it to D-glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this splitting is very important. In the next part of the pathway, D-glyceraldehyde 3-phosphate is run through a series of steps, and then which end in pyruvate. there is an enzyme called isomerase that takes dihydroxyacetone phosphate and converts it into D-glyceraldehyde 3-phosphate. This enzyme can go both ways. It can convert back and forth. And the cell actually pushes the equilibrium towards D-glyceraldehyde 3-phosphate because it removes the D-glyceraldehyde 3-phosphate, and then the equilibrium goes towards it, away from dihydroxyacetone phosphate. And the DHP is converted into D-glyceraldehyde 3-phosphate because of this. Remember when we talked about basic energy concepts, we talked about how cells can take advantage of equilibrium by removing products? This is one place where they do it. Okay, so more EMP. Now, the third part of this pathway is energy extraction. And one really important step is the first one here, right here. D-glyceraldehyde 3-phosphate is converted into 1,3-diphosphoglyceric acid. So you add 2 phosphates. This addition of inorganic phosphate does not come from ATP. It is energy. It is a free reaction. It's favorable. You add a phosphate, you actually get a reduced NADH out of it. This is the key energy generating step of the whole pathway, because now you've got 2 phosphates on here and you can go through several conversions, eventually making pyruvate, and in the process generate 2 ATP. So a phosphate is added for free and then 2 are peeled off. Since this pathway gets run twice, the net is that you put 2 ATP in and you get 4 ATP out, so 2 net ATP. Now in the Entner-Duderoff pathway that is shown here, it actually only nets 1 ATP, and you can see that you take glucose, you make glucose 6-phosphate, but then you go through different steps and eventually you end up with 2-dehydro-3-deoxy-D-gluconate 6-phosphate. Say that 3 times fast. One of those gets split off to make pyruvate. The other one is converted into glyceraldehyde 3-phosphate, and that goes through this process again. You generate 2 ATP here, you use 1 ATP here to charge the molecule, so you only have a net of 1 ATP. And you get 2 reducing equivalents of NADH. Now the nice thing about that is that those can actually be used by aerobic organisms, as you'll find out later when we talk about respiration. So this is a pathway that's actually advantageous for organisms that go through and do respiration. Okay, now the cell has gone through, they've generated a little bit of ATP from breaking this thing down to pyruvate, and pyruvate is a central metabolite, by the way. Again, you can see that here. Here's pyruvate right here, and this is a central metabolite. Now, we've gotten a little bit of energy, so we've got some ATP, we've got some NADH, but the cell needs a lot more ATP than it needs NADH. And in fact, the NADH is in excess. In fermentation reactions, you have to get rid of the excess NADH. If you're not using a respiratory pathway, which can burn up your NADH and make NAD again, you've got to get rid of that NADH and then recycle it for the next steps in breakdown of more substrate. In fermentation reactions, they get rid of this NADH to form the fermented end products. And it just so happens that, lucky for us, we like these end products. Okay, so fermentation. The definition of fermentation is an energy-yielding process whereby organic molecules serve both as electron donors and electron acceptors. There are numerous types of fermentation that have been found in the microbial world. We're just going to go through a few of the very common ones. Okay, so let's talk about fermentation. Fermentation is an energy-yielding process whereby organic molecules serve as both electron donors and electron acceptors. There are many types of fermentation. We're going to go through a couple, but this is found all over in the microbial world. NAD in fermentations is almost always reduced to NADH, and this is a problem. You've got to get rid of that NADH. Pyruvate is an important intermediate, and it is normally converted to something else. So here's some other general concepts. Energy in fermentation is derived solely from substrate level phosphorylation. Energy yields are usually low. You only get 1, 2, or 3 ATP per substrate oxidized. Oxygen is not involved in fermentation. It is a completely anaerobic process. Typical substrates for fermentation include sugars, amino acids, and typical products depend on the substrates. Organic acids are produced, lactic acid, acetic acid, alcohols, ethanol, methanol, butanol, or gases, hydrogen gas, and CO2. Okay, so let's look at some important fermentation. First of all, we're going to cover alcoholic fermentations by Saccharomyces, yeast. In this case, glucose is converted into 2-pyruvate, 2-NADH, and 2-ATP. And then the NAD is restored by producing 2-ethanol, 2-CO2, and the oxidized NAD. In homofermentation, lactic acid is the only product. It's important in food production, such as yogurt, and in our muscles if we're running really hard and we're not getting enough oxygen into our blood, then the muscles will produce lactic acid. In this case, glucose is converted into 2-pyruvate and 2-NADH plus 2-ATP, and then to restore the NAD, you produce 2-lactic acid and 2-NAD. Finally, there's propionic fermentation by Propionibacterium, and these are done in Swiss cheese. In this case, 5-glucose are converted to 3-pyruvate plus 3-NADH, and then to restore the NAD, 2 propionic acid are made, 1 acetic acid plus CO2 plus NAD. So those 5-glucose end up producing 2 propionic acid, 1 acetic acid, CO2, and NAD. All right, let's look a little bit more specifically at ethanol formation. This is a two-step reaction. Yeast will take glucose, take it through glycolysis, and produce pyruvate. So they get their 2-net ATP from there. And then that pyruvate is converted into acetaldehyde, and then the acetaldehyde is reduced to ethanol, and in the process, oxidizing NADH to NAD. Yeast such as Saccharomyces cerevisiae do this, so does the bacterium Zymomonas mobilis. So, why should you care? One example, and since we're in Wisconsin, it's a great one, is beer. Beer brewing is a huge industry in Wisconsin. It generates $722 million in 2002, and that was the last statistics I could get, and you can actually look it up on the census site for the government. There are at least 57 commercial breweries in the state, and some people think beer is why civilization started. So if you want to read more about that, here's a fun link you can go look at. So, brewing, what is it? Brewing consists of taking malted barley, water, hops, and yeast. That's the process, those 4 things. So what happens in the brewing process is you harvest the grain, it's usually barley, you soak the grain in water, causing it to germinate. You then dry and crush the grain, forming the malt. That malt is then mashed, and in the mashing process, what you do is you release the enzymes in the barley, and they convert a lot of the polysaccharides into maltose and other sugars that are then available to yeast cells. You add hops, which gives this characteristic flavor to beer, and also it has disinfectant properties. Yeasts are then added. They go through a glycolytic fermentation process, producing ethanol. You age the green beer for a few weeks, you finish and package the beer. Okay, so that's the whole process. And the important thing that we're talking about here is the fermentation that happens when the yeast are added. Another example of fermentation is lactic fermentations. In homofermentation, as we said, you take glucose, you convert it into glyceraldehyde 3-phosphate. This then goes to pyruvate, you make 2-net ATP, and then in a single-step reaction, pyruvate is reduced to lactate, oxidizing NADH to NAD, which can then go back around and react with more substrate. Lactic acid bacteria are the ones that perform this reaction, organisms like Streptococcus, Lactococcus, and some Lactobacillus. Heterofermentation is another lactic fermentation process, and in this case, you go through this conversion where you take glucose You convert it into ribulose 5-phosphate, right? And then this goes to xylulose 5-phosphate. It splits into acetylphosphate and diglyceraldehyde 3-phosphate. The diglyceraldehyde 3-phosphate goes converted to lactic, just like in the homolactic fermentation. But then you have this unique pathway. You make acetylphosphate, acetaldehyde, and then ethanol. And again, you have these conversions of NAD to NADH to NAD so that it's available to do work. And this process is a yield of only 1 ATP per glucose formed. Organisms that form this are Leuconostac and some other lactobacillus species. But as you can see, this is an amazingly inefficient process. These organisms chew through a lot of substrate to make just 1 ATP. Okay, now, it just so happens that we like this process. Lactic acid bacteria are used to make all sorts of things. And the main components of cheese and yogurt is cow's milk. Cow's milk is 3% fat, about 3.3% protein, 4.7% lactose with a pH about 6.6. These lactic acid bacteria can take this lactose and ferment it. And when they do, they produce lactic acid, which gives the milk characteristic qualities. So in cheese making, you collect and pasteurize the raw milk to then eliminate pathogens. You add a starter culture and you ferment the milk. That is fermented, and then a protease called rennet is added. It makes these milk curds. So you get like this slightly firm cheese, and from that you can make the fresh cheeses like cottage cheese. Other times that curd is concentrated and pressed into molds and salted, and then aged for long periods of time. And this can be for weeks or months or years. And one establishment I go to is advertising 10-year aged cheddar. I don't know if that makes a huge difference, but it does change the taste. And then the cheese is then removed from the molds and packaged for sale. Okay, so here's an example, milk fermentation. They're pouring it in the fat. The curd, you can cheddar the curd, right? You incubate it, it grows, and it ferments. And then you break up that curd. You press that curd into molds. In some cases, the cheese is then put in salt to brine it, and then it's allowed to age for long periods of time. Okay, another lactic fermentation is a vegetable fermentation, the formation of kimchi. Kimchi can also be spelled as shown here. It is, again, it's a fermentation of cabbage by lactic acid bacteria in coliforms. Lactic acid is a major product and gives it its natural flavor and texture. Spices in kimchi also give it its distinctive flavor. And kimchi is a big deal in Korea. This is a staple of their diet. They eat a lot of kimchi. Kimchi has been around for millennia. It's first referenced in 2600 to 3000 years ago in Chinese poetry book, and it was a recipe. And that recipe was put as a poem so people could remember it. There are hundreds of known varieties of kimchi, and it was originally just cabbage and beef stock, but spices were introduced with European contact in the 17th and 18th century. As I said, it's a staple of Korean cuisine, and the most popular is baochu, and that uses Napa cabbage, and the ingredients are Napa cabbage, scallions, red pepper, and ginger root, and here it is shown down here. But there's, as shown on the right, are a bunch of different varieties of kimchi that are made. The fermentation in kimchi is not a single organism that's added to milk that has very few microorganisms in it to begin with. It is a natural fermentation. So the natural organisms that are on the Napa cabbage when it's prepared are the ones that ferment. And the first thing you get is this wave of coliforms. Non-pathogenic Klebsiella and Enterobacter come in, and they will ferment the sugar to acid products and lower the pH to about 5. At this point, they inhibit their own growth. A secondary fermentation then occurs by Leuconostac, and these are lactic acid bacteria. They're heterofermentative, and they produce gas, and they stop fermenting at about pH 4.5. So they add more acid, and they drop it down, and then at this point, more acid-tolerant bacteria take over. And the last is the lactobacillus. They are homofermentative. They produce more lactic acid and no gas, and they will lower the pH to near 4. And if you look at this kimchi fermentation over a time of days, and they'll be fermented for different times, but I just picked about 12 days, you'll see this wave of coliforms come up and then drop. And this is, again, their viable count in the kimchi. You'll then see leuconostoc, maybe leuconostoc mesoenteroides come up and then drop. And then by the 4th day, the lactobacillus take over and they will drop the pH down to 4. Finally, the last example we're going to use is chocolate, which is called the food of the gods. It is a natural fermentation, and there's about 2.5 billion kilos of chocolate fermented per year. One of the reasons that it's so popular is it actually has 2 psychoactive products in it. Theobromine, which is a stimulant, and 2-phenylethylamine, which is an antidepressant. So eating chocolate can actually have psychoactive effects. Chocolate is, again, a very complex form of fermentation. Cacao pods have seeds, beans, that are covered in mucilage, which is carbohydrates. The pods are broken open and natural microbes ferment the mucilage. And then there's a complex succession of microbes make the products. And again, these are natural microbes that are on the cacao pods. Right? You'll have yeast and lactic acid bacteria come up first. They will do their fermentations. When the pH drops, their numbers will decrease. You start to see spore-forming bacteria increase, and then they're the last ones that are left. Acetic acid bacteria come up. They produce acetic acid, and then their numbers drop. And this is about a 1-week fermentation. Okay, now, these are just a few examples of the different kind of things that you can find that are fermented, but there are lots of other fermentations. There are alkali fermented foods. Vegetables, the fermentation leads to the release of ammonia and it raises the pH. Usually this is a bacillus species. Natto, which is a Japanese product, is a soybean product. Dawa dawa, which is West African, which is fermented locust beans, which is shown right here. And then there's the weird thing of making a 1,000-year-old egg, where you take a duck egg and you coagulate it using sodium hydroxide. Then you bury the egg for several months and then add bacillus, which leads to the transformation into this thing. My question is, A, why would anyone want to eat this? And B, who came up with this process? Hey, you know what? I'm going to make something new. How about we take an egg and we put it in sodium hydroxide and then bury it in the ground? Sounds like it would be really good. But hey, food's food. Another microbial process before we end this lecture that I want to talk about is involved in food production, and that's vinegar or acetic acid generator. You will take an alcoholic fermentation. So you'll take a product and you will ferment to ethanol, and then you will throw it into an aerobic process. And this is a vinegar generator where you squirt the alcoholic juice on the top. It goes through beechwood shavings, and there's an oxidation, and at the bottom you get vinegar out of the bottom. I know, so what's going on in this case is acetic acid bacteria use respiration, and they will convert this ethanol to acetaldehyde and then acetic acid, and in the process generate energy because they're using oxygen as the terminal electron acceptor. So this isn't a fermentation per se, but it is a microbial process that produces acetic acid, which is vinegar, and vinegar is a very popular product that people use in all sorts of foods. Okay, to end this, here's a whole bunch of other examples of fermented products. Some of them you've probably consumed yourself, and you may not have realized that they're fermented. So I just thought I'd end that with showing you all the different types of fermentations that take place. Okay, that's it for this lecture on fermentations.